join me to welcome Gabriele Sales.
So thank you, everyone. Thank you for the nice introduction. Really glad to be here today. So as you've heard, my name is Gabriele Sales. I'm actually working at the University of Padova as a computational biologist. Now, I guess I know already what some of you might be thinking, and that's kind of a pain point for me. What has biology to do with HPC to begin with? It might look like it's a kind of a disconnected type of application, right? So indeed, if you think about it, there are other type of research fields which have a much longer history of using this type of compute resources, and typical examples that come to mind include high-energy physics or maybe astronomy. And what's remarkable about these fields is the type of experiments that they put up. You can find some pictures here representing kind of the most famous ones. They not only produce huge amounts of data as they run, but also they require a lot of money, actually, to be run. In the context of biology, more in general, this is the kind of typical instruments that you might find around. These are sequencers. And as you will notice from the description here, from the price tag next to that, indeed we are working on a little bit of a different scale. But don't be fooled by the fact that these type of tools produce a modest amount of data in comparison to the one before, because if you go to a research center, typically you will find not one, but many of these humming along and working all day long, okay? So you don't have to consider any one of those in particular, but rather the aggregate output of all of these research centers across the world to really get a feeling for what's the general production. The point which I would really like to drive across today is that this different approach to data collection, being much more distributed geographically, has a profound impact in the way in which you deal with data itself and later on with its analysis. But before I actually get to discussing that, I guess I have to provide a little bit of an introduction to why we need these tools in the first place. So let's start from the typical example, that is genome sequencing. And since the end of the Human Genome Project in the early 2000s, people have got the final confirmation of the fact that indeed within our genomes are the information which encode for our own, say, physical appearance, our height, the color of our skin, things like this. And not only that, however, people are still learning a lot of this, right? But we already know that there are other traces within our genome that defines, for instance, whether we are particularly susceptible for diseases. And in the unfortunate event in which one of these actually appears, having a good grasp of that person's genome really helps in tailoring some sort of a therapeutical intervention for that. So, as you can imagine, there's a lot of interest there. No matter what your question is, whether it's basic research or whether it's clinical applications, in order to sequence one of those genomes, you need to go through a series of similar steps. Starting with, of course, the extraction of DNA from your samples, from your cells, but then you immediately need to take action and to break that down into smaller pieces. Now, the reason for this is actually kind of obvious in retrospect. Rather than dealing with one huge problem, you much rather work on a number of smaller problems, each of which is a little bit simpler to tackle. Not only that, but by physically dividing these objects into multiple parts, you can actually process all of them in parallel and therefore gain speed and throughput in the process. The drawback, however, is that once you've done that, the type of measurements that you get, every single one of those is no longer representative of the whole, and you need to put some effort in order to reconstruct the whole thing. These measurements are typically called reads, and as you can see here, you need a computational approach in order to connect them back, or if you prefer, to map them back to the correct spot within the genome where they belong to. And once you've done that, you will start going through the data, and you will notice that every now and then, while your reference genome, which, if you remember, was sequenced a few years back, tells you should expect one particular letter, in your data for that other person, you find something different, a mismatch. Now, of course, you shouldn't be trusting one single measurement for that, and eventually you will end up with something like this. This is called a pile-up, because you have a stack of multiple reads for the same genomic locus, which are essentially repeated measurements. And as you can notice here, in the column highlighted, there is some conflicting evidences. Sometimes you find G's, sometimes you find A's. And really, there are two possible explanations for this thing. It could either be the case that indeed you have found a biological variation of some sort, or you got into some sequencing error. Indeed, sequencers are not exactly ideal machines. They tend to make their own sort of errors. The actual figure will vary a little bit depending on the technology that you're using, but you might expect of making one mistake every 100 nucleotides that you sequence, which is not a small rate to begin with. So in order to distinguish true signals from everything else, you need some statistics. And you don't really have to think too hard about some tools for solving this problem, because you can reach for one of the oldest kind of techniques or results that were developed in statistics, the bias theorem that you can find here. And what's remarkable is how nicely it applies to this problem, because it allows you to compute the probability of a given genotype, that is a series of nucleotides, given the data that you have actually experimentally observed. Now, of course, I don't want to dwell too much on the technicalities here, so suffice to say that it is possible to convert that into some code, which you will find listed there. It's really, really short if you think about it, and still it works quite okay. Indeed, if you take this approach and you apply it to some real data for which you know the ground truth, you will find that the output of this method has a high concordance, a very high concordance with your expectation, more than 99%, which is quite good. However, as you keep looking into the other performance metric, that would be specificity, well, you discover something not as good. Indeed, this method will output a huge number of false positive results, and this really makes the entire thing unusable in practice. The problem, you see, is not really with the bias theorem, which is, of course, absolutely correct. Rather, it's the way in which we have applied to this particular problem. Specifically, sequencing errors are not IID by any means, and therefore they are biased in their own ways. If you really want to build an appropriate model, you have to take that into account, and that means complicating the model itself. And so people over the years have been working on this. They made some breakthrough, which allowed them to improve their performances, and still, in the end, they had to resort to something else, to some heuristics, which means, simply put, ad hoc filters for throwing away data when the quality in a certain sense is too low. You might think, oh, well, is this really necessary? So let me show you why that's the case. This plot over there has the false positive rate on the x-axis, the sensitivity on the y-axis, and therefore the ideal algorithm would actually live to the top left corner. The two curves that you will find there are actually two variants of the same approach. The first one, the dashed curve, actually is when you don't use those filters, while the continuous one actually corresponds to when you enable them. And you can appreciate how much of a gap is between these two things, and in particular, how the latter is actually way closer to the ideal. So imagine if you really want to inform clinical applications, you better use those filters in order to improve your chances of getting the right signals. This is a little bit frustrating, however, because a biologist looking at those filters will, well, find them quite reasonable, but then it might start wondering, why exactly do we need those and not many others that one can think of, which might look on their face like equally interesting? And the answer is that people have actually tried with many different alternatives, and these ones were working better on some real data, okay? Nothing more profound than that. So a few years back, some researchers actually were thinking about this problem, and were trying to come up with some alternative approach, which was a little bit less arbitrary and more principled, if you like. So I guess they were looking at another pileup like this one, and as it's customary, you can find here different nucleotides being colored in different ways, which is simply a graphical way of making them stand out, okay, on the screen. But they thought it's not really necessary to draw the entire letter on the screen, because just as well we could summarize that signal with one single color pixel. It would convey the same meaning, actually, if more compactly. So it's not difficult to implement this kind of an idea with a little bit of code. You can see Python code, actually. This is not pseudocode, by the way. It's actually some runnable Python code for implementing that. And indeed, in order to define one of the pixels, you need 3 numerical quantities, one value for the red, from a green, and from a blue channel. And if you really want to get fancy then, you can add a 4th thing, which is the opacity level for the resulting pixel. Now, in this code, to be honest, there are a few function calls, but they're not difficult to implement either. This is the full printout for the whole thing. And you might recognize, for instance, in the second function there, that the connection between these nucleotides, A, C, G, and T, and the corresponding shade of red, is actually established in the simplest possible way. It's a static lookup table mapping 1 to 1. So really nothing fancy going on here. When you apply this type of procedure on some real data, you get these type of plots. On top, you have a case for a genomic region in which you do actually have one of those mutations, which graphically speaking would correspond to that brightly stripe of pixel in the middle. The bottom panel, on the other hand, represents a different type of variant in which you actually lose an entire segment of a chromosome, which here results in that black rectangle at the bottom. It's remarkable, if you think about it, how even if you know nothing about genomes or genome sequencing, our eyes can immediately spot this type of graphical patterns in these plots. So, and here is what really makes the difference. We might actually try and make our machines recognize these patterns for us automatically. So what the authors of these research actually did, they reached for some deep learning methods, in particular one, which is quite popular. It's called Inception V2. And they actually trained this on those type of plots, adding a classification head that you will find to the right, which in the end essentially outputs one probability value representing the confidence of the model about the fact that these plots actually do correspond to a true mutation rather than being a false positive. And when they tested the performances of all of this, they actually discovered something quite difficult to really justify by first principle. This method, which is called deep variant, here highlighted in blue, actually managed to outperform even the state of the art back then, another software called GATK, which was based on more classical, like statistical and machine learning approaches. This was remarkable because it was one of the first examples of how you can take a device, a computing device for a completely different application like computer vision, and in a certain sense transplant this mechanism to a new domain, retraining from scratch, notice without changing overall the architecture, and still get state-of-the-art results. Many other examples have emerged in the literature. It was a little bit difficult to pick some of them because there are so many, so this is a completely arbitrary choice on my part. The first one on the left is likely the most popular. It's taken from the third release of the AlphaFold method, which is a computational method by DeepMind, part of Google now, which addresses a very thorny problem in computational biology. You want to predict the 3D structure of proteins starting only from their sequence. Now, this is a very complicated problem, but it's fundamental because the 3D structure of a protein is actually defining how that works at the molecular level. To the top, you will find another application. Now, these are graph neural networks, and they were implemented in order to reconstruct or to deal with, if you prefer, the typical heterogeneity that you will find in complex tissues. In all sorts of cases in which you have many different cell types which are interacting with each other because of their spatial proximity, and you want to sort them out, their identities. To the bottom right, you will find, well, our own contribution to the field, and here we had yet another different problem. In our case, we're working in collaboration with the Botanical Garden of Padova, which is arguably the oldest one in the world. And over there, they store a huge number, like really millions of dried samples of plants, which are kept for historical reasons. And now that we have the means, we would really like to go back to those and sequence their genome to understand how plants are changing over longer time scales. Problem is that some of these samples are really, really old, like 200 years old. So it's not really something you can take for granted that the DNA inside them is still of sufficient quality to actually do this sequencing. So what we would like to do is to have some tool for predicting whether you have some confidence, okay, in your capability of getting a good result out of that sample. So what we did, we take high resolution of images of those samples and we feed them into a convolutional neural network. We have some metadata about those, where they were collected and when, and we combine these two modalities of data with a transformer model to provide in the end one prediction. And we managed to actually make this work reasonably well. For all of the potential that this application actually seems to imply, there are still a lot of dark corners, however, in the use of these tools. Many times you find yourself wondering why that particular architecture is so successful at one problem, okay, and why not many of the others that have been proposed in the literature. And we are certainly not the only one. The irony is not lost on me that no longer than 2 weeks ago, this paper was published by a frontier lab in AI, which is Anthropic, and they actually propose, like they took inspiration in this strange cross-pollination between different fields to what neurobiologists have been doing for a very long time. So they went back, they took inspiration from their tools, and now instead of studying real brains, like living matter brains, they apply the same techniques to neural networks in order to understand how they activate different parts of a network. For instance, when they're telling the truth to your answers, or where they are making up excuses, or they're hallucinating, if you prefer, in order to just come up with something reasonable, okay, as an answer to your query. I think there's still a lot to be done here, but there is another kind of consequence of this shift, which is closer to today's theme. That has to do with how we deal with data collection, because you see, all of these methods tend to work well whenever you have sufficiently large data set, and typically the larger, the better. So the pressure on generating more data, if anything, has become more stringent, more strong than before. And to a certain extent, in biology, we're not in a tight spot, because over the years, we actually managed to improve techniques in order to really cut on costs. Just to give you a very quick comparison, that first genome, okay, I was speaking of, that was sequenced in early 2000s, costed something like $1 billion to get to the draft level. Nowadays, you can get another genome for less than $100. So it's a completely new result, which totally shifts the balance between how it costs, how much does it cost to gather the data versus what you need to pay in order to process that. So a lot of people are starting to think that maybe it's not totally economically sensible any longer to collect the data in one place, ship it from a network to another place, the data center, where it has to be analyzed, and then ship the results back to the source, because all of this back and forth actually costs time and money. And don't get me even started on the fact of how much does it cost in proportion to store this data for longer periods of time, which sometimes you're forced to do because of regulatory reasons. Indeed, I'm not the only one really noticing this. One of the largest databases for this type of data, the Sequence Read Archive, is a US kind of project which is backed by the NIH, recently shifted the policy for sharing the data with you. They're no longer giving you access to the pristine format of the data, rather to a light version, which is essentially a heavily quantized transformation of that, which is a fraction of the original. If optimizing storage, okay, and data distribution is a nice thing to have, it's not like it's really opening new type of directions for your investigation or new type of techniques. So let me make an example of something which is really a game changer then. And for that, I would like to go back to the problem of this sequencing. So these mutations I've been talking about tend to be, and hopefully so, rather infrequent in, well, everybody's genome. They actually appear once every 100 million nucleotide on average. So at the measurement level, this is kind of a problem, because since when you do genome sequencing, you essentially sample randomly different positions within the genome, you have to try really, really hard until you eventually manage to find one spot corresponding to one of those mutations. Most of the measurement that you make, it's not like they are wrong, but they do perfectly correspond to your expectations. So they don't contribute nothing new. For a very long time, this was the best that people could do, so they really had to repeat this measurement multiple times. More recently, a new generation of sequencers has emerged, like this one. This is called a nanopore sequencer. And the name actually comes from the structure of it. It actually works by making these DNA molecules run inside a small pore, or a nanopore indeed. And as this process goes on, the DNA molecule will interrupt the current, or will alter a current which is flowing across the extremities of this pore. And if you measure that as a time series and you feed that in an appropriate neural network, you will manage to reconstruct the identities of those nucleotides. But look at what's happening here. If you take that current and forcefully you change it, then you would be able to invert the procedure. Rather than pulling the molecule in, you would actually push that out, and rather quickly. In other terms, what you manage to implement by this way is some sort of a filter. You can accept or reject different molecules at your will. So people have been trying to come up with some ideas, creative ideas for exploiting this process. So whenever you want, for example, to sequence a genome, we can start by a shallow pass, and we accept every molecule that is coming by, and we start taking measurements. And it will gather some kind of initial evidence of the fact that in different positions within these chromosomes, you actually potentially have some mutations. From that moment on, you can actually start biasing your selection procedure, okay, so that you will focus on certain particular spots, like, for instance, that one. Essentially, what you will do, if your molecule potentially belongs to that region, you will accept it. Otherwise, you will throw everything away. So in the end, you will manage to improve your signal-to-noise ratio, and therefore you will become much more efficient at really getting the signals you're looking for. Problem is, all of this only works whenever you have a lot of compute power. And indeed, these machines are actually sold in conjunction with servers which are equipped with very powerful GPUs, but as the authors of this method actually mentioned, this only works up to a certain level, and indeed, they ran this approach for small genomes. When we tried and scaled that to larger ones, and indeed our own is quite a large type of genome, this actually fails. It's too slow to be of practical use. And notice that here there is something which is kind of subtle. This application that I'm talking about here is drastically different from all the others, because here you have to proceed in real time. It's not like you can store everything and process later, because part of the loop is you want to control the instrument itself as it's progressing, because these choices are irreversible, whether you want to accept or not one molecule. So what this is compounding to a certain extent your problems is that it's not only a matter of throughput, it's also a matter of latency. You have to react real quick, because one of these pores is actually sequencing around 400 different nucleotides per second. So it's not like you can stay there and think about it for way too long. And therefore, if you were to connect somehow an instrument like this one to a data center 100 miles away, I would guess that the lag introduced by, the latency introduced by all of this would make your response time too slow to be really usable. So the only option left then is to take your compute power closer to the instrument. It's not exactly near to that, but at the very least in the same building. And this is introducing a lot of infrastructural challenges, because now you're addressing some of the same needs of a data center, but in a building which was never designed for that purpose. There is another example of a new challenge, okay, which is showing a little bit the cracks in the typical approaches that we have been using up to this point for dealing with computational biology. And that has to do with a different type of an application, a different type of novelty, okay, in the way in which we process our sample, that is single cell gene expression measurements. So the idea is that when you want to measure how different genes are being used in different cells, what you do is, again, a slightly different kind of sequencing, and you reach for a bunch of cells and essentially you smoosh them in order to extract the interesting molecule, okay, for your own purposes. But in doing so, you're actually mixing the contribution of different cells, which at a very profound level may actually be of different kinds. So you get some sort of blurred out version of your original data. More recently, in the development of molecular biology, people have been gaining the technical tools for dealing with a minuscule amount of molecules that you find within single cells. And now they can track all of those one by one, which increases by a huge amount your actual resolution into all of these differences. So what you end up with is essentially a matrix like this one, in which you have different genes on the rows, different cells on the columns. And again, just by looking at these graphical patterns, you will immediately notice that over here you have 3 different groups of cells. And those gene names to the right will speak volumes to immunologists, for instance. They will recognize that these are cells from the immune systems, quite famous cells, T cells and B cells. Okay, they're quite fundamental building blocks of our immune system. There is a problem, however, with this type of representation. Eventually, you will run out of pixels because as you keep increasing the number of cells, it's only so much space that you can find on a given monitor. So you would rather represent the same information in a somewhat more compact way, and for that, we can resort to dimensionality reduction techniques. One of them, I guess everybody is familiar already with, is the PCA, or principal component analysis. It's one of the oldest, and it's a linear method. Unfortunately, this type of approach tends to struggle whenever your problem is highly dimensional, and guess what? Gene expression is actually one of such kind of problems. So people more recently has actually developed alternatives which are non-linear in the way in which they proceed. One of them is very, very popular. It's called the UMAP. So now biologists equipped with this new toolkit for dimensionality reduction actually managed to perform experiments like these ones. So in this case, what they did, they extracted cells from all sorts of different organs within the body of an adult mouse. And when they take this measurement and project them in the UMAP that you will find on the right, over there you have one dot for every one of those cells. And you will recognize that they tend to cluster in this reduced space in a way which closely mirrors the original organization in different tissues, in different organs. So what we'll learn in the process is that indeed different organs are really responding or being regulated by very different gene programs, one from the other. And imagine the amount of information now that you can extract from these plots in order to understand how these cells come to become so different one from the others. Because again, they all share the same genome, okay? So the base information is exactly the same. It's only the way it's being used dynamically from one spot to the other that changes. This was just one experiment, though. Over the years, many more have been accumulating in the literature so that nowadays we have access to databases like this one, harboring more than 100 million cell profiles that you can query. So it's only natural that people nowadays are starting to think about what's the most efficient way to sift through this data. And we've been working on this problem too, with some colleagues, Chiara Romualdi and Davide Risso, who have been starting by benchmarking some of these applications and finding how we can build an entire pipeline out of different pieces, which is as efficient as possible. These pipelines indeed are made of many different stages, and you might recognize that one of them is indeed our good old friend, the PCA. For such a well-understood problem, you might expect that the implementations that you find in software are all kind of optimal. Well, we're actually quite far from something like that, because here every bar that you will find represents the runtime of a different implementation, different algorithms, different memory layout for storing the information, and there's a lot of variability. Mind you, the y-axis actually is in a logarithmic scale, so really you have a kind of a one-order difference between the best and the worst in that particular respect. But I don't want to kind of focus too much on the plot itself, rather on the story and how we got to this particular point, which is, I guess, more interesting to us nowadays. So, as Ilaria Billato, a PhD student working on this problem, started actually developing this benchmark, she was working on her own workstation, and there, one of his stages actually took 64 seconds to run. Imagine how unsettling it was when she actually moved the same data and the same code to a much beefier server, and there, everything slowed down, more than 3 times as slow as before. So this really took way longer than I would like to admit to really understand what was going on there, and eventually it was something kind of lame. We discovered that on the server, actually, the version of BLAS being installed was the netlib reference version, which, as you might remember, is not exactly the best tuned for modern hardware. So what we had on the workstation on the other end was OpenBLAS, which is much fitter for that. So when we actually switch between these two on the server, we gain back our performances and some more. Now, you might think that, well, this was an unfortunate accident, and really on a high-end system, you have a kind of knowledgeable system administrator. We have already taken care of these aspects. But, and that's the interesting part to me, we've been stressing a lot on the fact that people should be using reproducible means of research, which, technically speaking, it eventually boils down to containers or isolated environment of some sorts. So what people typically do is they develop their own software, and eventually they embed that within a container, which is based on some sort of a common base image provided by a major Linux distribution or something like that. And guess what? Which is going to be the last version which you will find by default within one of those? It's again netlib, okay? So again, no matter where your container is going to run, no matter how highly tuned that particular system is, because this is a container and it is isolated, you will find the same problem, okay, if you're not aware of all of this. So you risk running some things which are not really making the best usage of the available resources. And to be honest, this is not even the worst part of this all. Indeed, as we were kind of trying and address this problem in particular, we end up doing a lot of performance profiling. And eventually what we found is that this was, essentially this one here, was the hottest expression in our whole kind of computation. If you look at that, it's actually quite easy. It's simply a multiplication followed by an addition, which is eventually an accumulation. So if you look at the disassembly, you will find that these two instructions nicely match that expression, and you might find this reassuring, but actually it's not, and it's not at all, because if you have been following the evolution of hardware, you will remember that since these come so frequently together, people have devised specific hardware instructions for tackling them both at the same time. They're called FMA, or fused, multiply, and add. So in the x86 world, this is what you would really like to find in your code. And the reason why we didn't really get those in the first place, it's again defaults. Typical Linux distribution actually do tend to favor, and for good reasons in general, at the very least, they do tend to favor compatibility with older architectures, and therefore they don't push compilers to really make use of the latest and greatest in terms of instructions. They tend to be conservative, and therefore they produce inefficient code. And to be honest, this is not yet what you would really like to run, because there is one extra step that you need to take. Now, this is a little bit smaller, but let me give you the gist. You really want to process more than one number in parallel. Indeed, you have large CMD lanes in modern CPUs, so why don't we use those and process in parallel 4 of those doubles or 8 of those doubles, something that you can do again with some more recent instruction sets. The take-home message is that once you do that, once you fiddle a little bit with your compiler, and notice, without changing any single line of your code, only working at the level of the compiler and its flags, you can manage on the same hardware to gain as much as 20% of performances, which is already quite a lot if you think that the initial code was already quite close to be memory bound. So I wasn't even expecting this large of an increase in performances. There is a problem, however. It's not like you can expect that every single user has the knowledge or is even willing of spending the time of recompiling everything from scratch, like I did here in order to generate these results, okay? So you might think of, is it possible to get to the same results or kind of there with alternative approaches? Well, one thing is runtime detection, for instance. You can see an example here. Eventually, as you go through your code, you can query about the features of your particular CPUs you're running on right now, and if that matches a given profile, you might actually dispatch your execution to one particular part of your code which is being more heavily optimized for that particular architecture. But you see, there's a drawback here because you have to re-implement from scratch your code every time a new architecture is being released. So it's a lot of work, it's continuous maintenance over time. So it's only natural that people right now are starting to ask for some level of support by their compiler vendors. And here you will find kind of a proposal for the Rust compiler addressing exactly this type of automation. But even then, and this was kind of funny when I read it, the author here, which is a compiler writer, so very knowledgeable about these details, we notice that only for one type of extension, AVX 512, you find in the wild 12 different configurations. So even if you had a sufficiently smart compiler, as they call them, imagine if that had to automatically regenerate, 12 different versions for the same code block. Imagine how large your binaries would become. So it would be a complete mess. An alternative approach I'm aware of is that of high or large-scale deep learning frameworks like Torch. And nowadays, they offer actually two different ways for running your code. One is eager mode. It's actually running on the spot. The other one is lazy. You start by tracing through the actual code, and you build a graph. And then you start essentially doing a JIT in the sense that you specialize those operations, and you try and fuse them as much as possible, one with the other, and again, to bring that as close to the hardware as you can really manage to do. This is, on the surface, much more powerful than before, but again, it has some limitations. First is, it has a lot of complexity, because now you're kind of bundling an entire compiler with your library, which is something that not really everybody can pull off. The other aspect is that this is not really working for all types of applications. Sometimes you will find that the time it takes for this machinery to actually specialize your code is actually longer than the original runtime for your piece of code that you were trying to run. So overall, this would be a net disadvantage to running the code, even if it's not optimized at all. So I can't really share one single solution for all of these problems. What I can propose, and this is something that I'm actually running on right now, is that we take some first steps, if not for finding the entire solution, but at the very least in the right direction. And I'm working here in collaboration with a bioconductor project, which is arguably the largest repository for bioinformatics software for the R programming language. And what we did here was to develop the infrastructure for, at the very least, try and make compiled code a little bit more optimized. Following what others have done, we are essentially subdividing existing architecture into major categories, and then we are targeting every one of those with as much optimization as we can, with the infrastructure again such that when the end user asks for a binary, it will receive the one fitting the most its own architecture. So this, I hope, it's at the very least one step forward. And just like you can think about CPU support for this, we're actually starting to build something for GPU code too. So in conclusion, I hope that the examples that I've been making kind of make the point across that a shift is going on in computational biology, and I think also in other type of applications. And indeed, for many years, we've been trying very, very hard to extract the last drop of performances from high-end systems. But in doing so, we've kind of neglected the typical hardware that we have in front of us, like our laptops or workstations. And it's like we're stuck in a previous generation because they're still using unoptimized code paths, And this is not just any inconvenience, because it risks creating a gap with a typical end user, which doesn't really feel appropriately the kind of resources that he or she has in front of himself, right? And therefore, how can we expect then these same users to really manage to scale up whenever they eventually end up using a high-end system? They lack really the conceptual tools to understand what's going on there. So I think this is a trend that we have to invert. And essentially, one first step would be to act on defaults again, because they are kind of important here, rather than hampering, and as they are doing right now, every attempt at making things more optimized. These defaults that we use should at least give you a nudge in the right direction in order to make things smoother for the end user. And again, this is something that I've been trying to work on. I've heard about other people working in this space, so if you're interested in this, I would be very interested in learning your own experiences and which type of approaches have you used in order to solve these challenges. With that said, let me thank you for the attention.
Okay, the mic's working. All right, any questions? Okay.
It's a silly question. So the bioinformatics stack, it's very difficult to compile from scratch, right? So basically, some of the tools that are used in a normal workflow require a given dependency multiple times because the code was not updated for years, right? So I wonder if it makes sense that instead of actually optimizing the compilation of all these versions of the application, if we can basically try to port them to let's say a new workflow of compiling and deploying software. Like, you know, you have things like SPAC that allows you to do things and compile on the fly. So that would allow us to actually manage better the stack, because today's are highly difficult. So it's not that we want to use containers for free, for fun. It's that, you know, sometimes it's impossible to compile this bioinformatics stack, right?
Yes, I think that that's a really interesting dynamic which has more to do with social interaction, say, rather than pure technical things. And let me go back here to this project, which I'm really interested in, because I would say more than half of Bioconductor has to do with supporting users and supporting developers too, in the sense that when you actually produce some code, what they will do is they will insert that into a CI system, and every 2 days they will rerun all sorts of tests that you have in there. And in the case that there's a regression, either in your code or in some dependency, they will actually notify you of that, and if you don't react, eventually they will nag you because of that, okay? So this is really important because it's all too easy. We are all busy, okay, in our lives, and therefore it's all too easy to publish something, and then you have your publication, you go on with your life, you forget about it, and that bit rots, okay? A lot of bioinformatics software is in this particular stage. Projects like this one really try and invert this thing because they actively try to actually help you in maintaining that. And sometimes, if you don't react because you don't have the time, you will find somebody else which is not typically the end user, but it's a more advanced user, which is not the developer of a package, however, that makes pull requests in order to solve the problem in the first place, okay? And sometimes, because of this project, people will apply this data in case you are no longer responding, okay? Somebody can actually become the new maintainer automatically for all of that. So yes, there is technique, but then there is also this kind of, again, of workflow, human workflow, okay, to deal with that.
Any other questions? Okay. Thank you, Gabriele.